Hello everyone, a very good morning to all of you. So today let's talk about nerve physiology. So nerve physiology is very interesting because see, imagine the sensory informations what we receive is sensed by the CNS system and we receive it immediately. Say for example, when someone touches me, the information is so fast that I don't even feel a lag in receiving the sensory information. So that action is taken care of by the neurons. So neurons basically carry the information, the sensory information or the motor information to the CNS and also receives the information. So basically, when imagine a person carrying a book to another area, okay, but in this situation the area is too long, that is the distance is too long, so we need several persons to carry the information to the destination. Similarly, the information what we receive should be carried to the CNS, but not one neuron can do it, so it has to be a cluster of neurons or a group of neurons carrying that information to the CNS system. So before moving, I'll tell you the basic parts of a neuron. Okay, so neuron as what is called as the dendrites. Okay, these dendrites, they carry the information. To be specific, they carry the or they receive the neurotransmitter. They receive the neurotransmitter. Okay, so they receive the neurotransmitter from the adjacent neuron, right? So these dendrites, they are basically a group of extensions which is present in the neuron that receives the neurotransmitter, whatever it may be. Say for example, glutamate. Okay, so the next one will be soma or body. So the body of a neuron, it carries a few important structures like the nucleus. Okay, so the information will be carried from the dendrites to the body and then it is carried to the axon. Okay, so axon, this is where the action potential takes place. We'll talk about everything in the following the following slides. So this is the axon. So the nerve fibers, if you see, myelinated nerve fibers, so it has myelin sheath. So as I talked in the previous videos, myelin sheath will have what is called as the nodes, that is nodes of Ranvier. Okay, so the action potential of a nerve takes place in the axon. So it is carried out and then again these dendrites, they transmit the neurotransmitter to the another neuron. So the cycle goes on until it reaches the CNS. Okay, so these are all the terminal axons. So okay, now moving on, before moving into what happens during the action potential, you should know what is called as the resting membrane potential. So what is resting membrane potential? RMP. Okay, so imagine a cell. So imagine, say for example, this is a nerve fiber. So usually if you see a nerve fiber as what is called as the pump, so in this case there is something called as sodium potassium pump. So the sodium potassium pump, it is going to bring 3 sodium ions outside the cell and 2 potassium ions inside the cell. So what happens? The sodium which is present inside the cell, 3 in number, is taken out and the potassium which is present outside the cell, 2 in number, is taken inside. So now you have to tell me that the charge inside the cell, will it become negative or positive? Obviously it is going to become negative. Why? More sodium is taken out and less potassium is taken in. So say for example, in this situation, let us assume the membrane potential, resting membrane potential, as 0. Okay, so because of this pump, the resting membrane potential becomes slightly negative. Okay, say for example minus 5 MB. Okay, now there is some other channels like what is called as the leaking potassium channels. So these leaking potassium channels, what it does is it takes the potassium outside the cell. See, always remember, when the sodium and the potassium pump causes more influx of potassium, so the concentration gradient of the potassium inside the cell increases. So because of the increase in the concentration gradient, via the leaky potassium channels, the potassium moves out of the cell, higher gradient to lower gradient. So while moving out, the potassium alone doesn't move out. The potassium carries or it binds to certain ions inside the cell, say for example phosphates. So phosphates are negatively charged, potassium is positively charged. So they bind together inside the cell. But while moving out, only potassium moves out, whereas the phosphate stays inside the cell because phosphates are a relatively larger molecule. Okay, so they stay inside the cell. So what happens? They leave out negative charged molecules. So what happens? The cell becomes more and more and more negative. So say for example, from 5 it goes to minus 90 megavolts. So initially, we are assuming it is 0. Because of the sodium potassium pump, more amount of sodium goes out, 3 sodium molecules goes out and 2 potassium comes in. So because of the change in the number, the membrane becomes slightly negative. So from 0 it goes to minus 5, and because of the concentration gradient, the potassium moves outside the cell via the potassium leaking channels. These are the potassium leaking channels. So it leaves the negative molecules inside and only the potassium moves out. So the membrane becomes more and more negative, that is it reaches minus 90 megavolts. Now there is another channel called sodium channels. So the sodium channels here, as I told you, outside the cell the sodium is going to be more, right? So 3 sodium is taken outside of the cell. So the outside the concentration gradient of sodium is going to be more, and sodium is going to enter into the cell, okay, because of the changes in the concentration gradient. So as this 90 megavolt becomes slightly less negative and it reaches minus 70 MB. So now the resting membrane potential of a nerve is minus 70 MV. So the resting membrane potential is controlled by 3 activities. One is the sodium potassium ATPase pump, where it pumps out 3 sodium outside the cell and 2 potassium inside the cell. The cell becomes slightly negative, and because of the concentration gradient, more potassium is present inside the cell, so it tries to move out to the area where it is present less via the leaky potassium channels. This is 2, this is 1. So it moves out of the cell, but while moving out it doesn't move out it alone doesn't move. It leaves the phosphates inside the cell. So the phosphates are negatively charged molecules, so the cell membrane so the membrane potential becomes more and more negative, and the sodium which is present more outside the cell, now it is going to come in via the leaky sodium channels, and it tries to compensate the negative potential. So the resting membrane potential of the nerve comes to around minus 70 MV. Okay, this is the very basics. You should never ever make mistake. So you understand these 3 concepts, you can easily correlate any questions related to this. So now imagine, when I want to stimulate a nerve, so say for example, if you want to make me angry, there will be some kind of threshold for me. Say for example, some people, even if we scold one word, they'll get triggered, they'll get offended, they'll shout, or they will whatever emotions they exhibit. But some, they require a lot of time to attain the threshold. That is, even if you scold the person for a more amount of time, only they'll get anger. So it varies. So similarly, for a nerve to stimulate, I should know the threshold potential of a nerve. That is, at what point it will get triggered. That is called as the threshold potential. So for the threshold potential of a nerve will be minus 55 MV. So when I reach this threshold potential, the nerve is going to shoot. It is going to go up. That is called as the action potential. The depolarization takes place, action potential of a nerve. So once the resting membrane potential moves to the threshold potential, the action potential is stimulated, depolarization takes place. So we will talk about everything in the upcoming slides. So moving on, as I told you, any membrane or any nerve, it has to reach the threshold potential to be stimulated. So imagine a neuron releasing neurotransmitter. Okay, it is a chain reaction, right? So neuron receives a neurotransmitter, resting membrane potential is changed to the threshold potential. Now depolarization takes place, action potential takes place, and then again it releases the neurotransmitter. The chain goes on till it reaches the destination. So the neurotransmitter is released. So this is called as presynaptic neuron. Okay, and the neurotransmitter is taken up by a next neuron, that is postsynaptic neuron. Okay, presynaptic and postsynaptic. Okay. So neurotransmitter, say for example glutamate, is released by the neuron, and what happens once the neurotransmitter is released, it is taken up by what is called as the voltage, very important, voltage gated sodium channel. So imagine a neurotransmitter is released, it is taken up by the voltage gated sodium channels. So what happens is more amount of sodium is released inside the neuron. Okay, more amount of sodium is released. So what happens? The resting membrane potential starts to change. So what happens? So at some point of time, minus 70 MB becomes minus 55. Once it touches this minus 55, as I told you, what is going to happen? The action potential is stimulated. So if you want to, a much clearer view, so imagine neurotransmitters are released. Okay, it is taken up by what is called as the voltage gated sodium channels in the dendrites. So you have lots and lots and lots of voltage gated sodium channels. So all these are going to pump in the sodium. So what happens? I'm going to get lots of sodium inside. Okay. So what happens once this sodium, it reduces the resting membrane potential. It reaches to minus 55. That shoots up what is called as the action potential. So the action potential takes place in the axons. So what happens is once the resting membrane potential is released, is touched, or it is reduced, it comes to minus 55. The more and more and more of voltage gated sodium channels open. So here there will be lots of voltage gated sodium channels. So more of sodium is released. So the cell reaches the membrane potential of plus 30 MB. So once it reaches the peak, what happens? It releases the neurotransmitter to the next neuron, and then it comes down. Okay. So once it reaches the peak, there's an absolute refractory period. I'll draw the graph. Okay, there's an absolute refractory period. It releases the neurotransmitter, and what happens? These sodium gated channels, they block. Okay, so what happens? These sodium gated channels, they get blocked, so no more sodium channel comes inside. And what happens? The potassium channels are opening and potassium channels are coming in. So potassium channels carry less charge compared to the sodium. At the same time, the sodium potassium pump is activated, so the sodium is taken out of the cell. So the cell again comes back to the resting membrane potential. So that is called as repolarization. So depolarization, release of neurotransmitter, absolute refractory period, repolarization takes place, and the nerve comes to the resting membrane potential. So this is what is happening in a nerve or a neuron. Okay, so see, say for example, I'm just drawing a graph. Okay, I'm just drawing a graph where the nerve is getting stimulated. Say for example, this is the membrane potential of a nerve. Okay, so it is 55 MB, and so once the nerve membrane potential reaches the threshold potential, say for example it is minus 70 MV, it is minus 55 MV, it reaches the threshold potential, there is going to be a sudden shoot in the nerve fibers. More and more and more of sodium is going to get released. So at some point of time, there is an absolute refractory period, and the potassium channels open, and the sodium potassium pump activated. So the nerve membrane, it becomes hyperpolarized, and then again the sodium channels closed, so it retains the resting membrane potential. So this is what is happening in a nerve. So this is the action potential in a nerve. So impulses carried out, the neurotransmitter, whatever it may be, touch, feel, temperature, proprioception, whatever it may be. So the impulses are carried, the neurotransmitter is released, the neurotransmitter is picked up by the dendrites, sodium channels are opened, and once it reaches the threshold potential, it triggers the action potential, more of sodium channels gets opened, release of the neurotransmitter to the next neuron, and then the potassium channels open, sodium potassium pump is opened, and the nerve gets hyperpolarized, and again the potassium channel closes, the nerve comes to the resting membrane potential. Okay, one important thing which you should keep in mind is the influx of the sodium channels, that is the opening of the voltage gated sodium channel by the neurotransmitter, causes the influx of more of sodium. So this is called as ESIP, excitatory sodium influx potential, okay, or excitatory stimulation. So what it does is basically ESIP, excitatory sodium inhibitory potential. So it causes more and more of sodium influx. So sodium influx is more, positivity is more, nerve reaches resting membrane potential. What happens if there is more of negative ion influx? Say for example GABA. GABA releases chloride ions. So more of negative ions are released, right? This is called as ISIP, inhibitory, excitatory inhibitory. So whenever there is ESIP, the positive ions comes in, action potential is stimulated. Whenever chloride ions comes in, like say for example GABA, so there's going to be hyperpolarization, not depolarization. Okay, these are very basic differences. So this is about the basics of a nerve physiology. So in the evening session, I'll be talking about a few tracks like spinothalamic tract, corticospinal tract, dorsal column pathway, and we'll be talking about the nerve fibers, A delta, A beta, C fibers. So we'll talk about how lidocaine numbs the nerve fibers. So see you all in the evening session. Thank you so much for watching the video. Have a nice day.